A very lovely good evening to all of you. I am Dr. Pooja, and today I will be discussing some last minute revision points for proteins. Now, in my last video, I had tried to cover most of the important points regarding the metabolism and regarding the general structure and function of carbohydrates. Since your exam is going to occur on 18th of this month, so what I want is, because whenever you are going to give your exam, you should always go through the last minute revision points, because it's not, it's very difficult to undergo, all the 19 subjects in detail. So last video we discussed about the carbohydrates, and today I will be discussing about the proteins. And in my video, I have tried to cover all those important topics, all those important points which have been discussed in your previous year examination. So just pay attention to this, and always whenever you are, learning something, just keep a copy and pen with you. Even though this is a recorded session, you can go through the video so many times, but always remember, whenever you write something with a pen, it is retained in your mind for a longer time. So try to, imbibe the important points. So without taking much of your time, let's start up with the last minute revision points for the proteins. Now see, indole ring is present in tryptophan. All of you know that tryptophan is a heterocyclic as well as a aromatic amino acid. Why it is aromatic? Because it has that aroma, or it has that benzene ring with it. But it has a special indole ring, which is a characteristic feature of tryptophan, and it is known as heterocyclic amino acid. And this gives a positive Hopkins-Colles test. A very important question which can be asked in the exam, that Hopkins-Colles test is specific for indole ring. Then next point, methyl donor in the amino acids is SAM. And you know what? This SAM stands for S-adenosylmethionine. Because all of you know that methionine gives rise to one carbon metabolism, or if we have to be precise regarding the function of methionine, it is an active methyl donor. And whenever it has to donate this methyl group, it has to be activated, obviously. So the activated form is this SAM. So you can remember, you must be listening to the songs from Sam Smith. So Sam Smith, SAM, S-adenosylmethionine. Then the proteins rich in basic amino acids which function in the packaging of DNA in chromosomes are histones. Again, very, very important. Always remember, if we look at the structure of DNA, DNA is always wrapped with histone. This is a very important question which was asked in the exam in the year 2017. How do we define histones? They are the basic proteins rich in lysine and arginine. And they are 4 in number, and they are present as an octamer. H2A, H2B, H3, and H4. And H1 is the linker DNA. And this is how they are present on the chromosomes, and they are also known as nucleosomes. So nucleosome is DNA plus histones. And histones are the basic proteins rich in lysine and arginine. That is what is written here in the slide. The next important point, an example of phosphoprotein is casein. Now, casein is the principal protein present in milk. Then sometimes the questions are asked on the classification of the amino acids. Classification mein you should know that how does an amino acid looks like and how. I'll just show you the structure. And I'll just show you one more important point about this. The generalized structure of an amino acid is this. This characteristic structure is known as zwitterion. And amino acids are amphoteric in nature. Amphoteric means they can behave as an acid and they can behave as a base. And the position here, the R group varies, on which the classification of the amino acids is based. So here it is acidic. So acidic amino acids are two, which are aspartic acid and glutamic acid. And one very important thing which I'm going to tell you here is this aspartic acid, its amide derivative is asparginine, and glutamine is for glutamic acid. So all of you know the importance of glutamine in the body. It is responsible for the transport of this toxic ammonia. So acidic, then if we talk about the basic amino acids are lysine, arginine, and histidine. Right? Then another important question which is asked in the exam is regarding the fate of carbon skeleton. So if we talk about the fate of carbon skeleton, we differentiate amino acids as per carbon skeleton. We differentiate amino acids as glucogenic, ketogenic, and amino acids which work as glucogenic as well as ketogenic. A very important question which is generally asked in the exam. So ketogenic may always remember leucine is there. Leucine is a potent ketogenic amino acid. Glucogenic and ketogenic are PITTL. PITL. This is a mnemonic. P stands for phenylalanine. I stands for isoleucine. T stands for tryptophan. This T stands for threonine. And this T stands for lysine. Lysine is predominantly this, whereas leucine is predominantly ketogenic. 1, 2, 3, 4, 5, 6. Rest 14 are glucogenic amino acids. Always remember this question can be asked in the exam. Right? The next, protonation and deprotonation at neutral pH occurs for histidine. Histidine is a basic amino acid which is considered to be very stable at intracellular pH. So deprotonation, removal of proton or addition of proton acts at the neutral pH for histidine. So slight change will disturb the entire osmolality. Right? Moving on to the second slide. Very important again. Always remember these last minute revision points are actually made to make you understand that these are the questions which can be asked in the exam. Creatine and glutathione, these are the true tripeptides. Tripeptides means they are formed of 3 amino acids. So mnemonic for creatine is GAM, GAM or JAM, whatever you can say, glycine, arginine, and methionine. Methionine is again donating methyl group here. And glutathione, which is a very, very important antioxidant, is synthesized from GCG, glycine, cysteine, and glutamic acid. So these two are the very important tripeptides. Creatine, all of you know, this is the energy reservoir for the muscles, and glutathione has so many roles to play. And this glutathione, I'll just take it in detail because glutathione is such a topic on which so many questions have been asked. One is obviously, which we discussed, it is a tripeptide formed of GCG, glycine, cysteine, and glutamic acid. Glutathione exists in 2 forms. This is oxidized and this is reduced. Both the forms are interconvertible with respect to one another. And then there is one very important enzyme, glutathione peroxidase. This enzyme consists of selenium. This was the question which was asked in the exam. Selenium is the trace mineral which is required for, which is the, you can say, component of glutathione peroxidase. Right. Then next is tryptophan, is a heterocyclic amino acid. Just now I told you because it has a special indole ring. Then proline is never found in alpha helix. Right? Proline is never found in alpha helix. Okay? Because it destabilizes the helix. Right? And then serinocysteine is known as the 21st. If we see, there are 20 standard amino acids. There are 20 standard amino acids. 21st is selenocysteine and 22nd is pyrrolysine. The only thing is that they are coded by nonsense codons. As selenocysteine hai, it is coded by UGA. But still it is counted as one of the important amino acid as it is required for the, as a coenzyme for so many important reactions. Right. Then moving on to the next slide. A very important slide. Always remember, one question is bound to come on the odor of the urine. You have studied the metabolism of amino acids in detail, so I'm not going to touch that. But yes, we will be discussing the disorders which are associated and what kind of urine odor is produced. If we talk about isovaleric aciduria, it is sweaty feet. This is a disorder of branchial amino acids. Phenylketonuria, musty odor. Maple syrup urine disease, the urine starts giving the smell of burnt sugar. In Hawkinson's-Urea disease, the odor of urine is swimming pool kind of odor. In multiple carboxylase deficiency, it is tomcat urine. In Osthaus syndrome, it is a syndrome associated with sulfur containing amino acids. It gives a hops-like odor. So these are what? These are some of the urine disorders which are associated by the following disorders. Right. After this, a very important thing which I am going to tell you here is, in phenylketonuria, this musty odor is due to the presence of phenyl acetate. This is important. This is again one of the very important questions which has been asked in the exam also. Right. Then we move on to the next slide. All collagen has a triple helical structure, a very important one. And triple helix it consists of the repeats of glycine, proline, and lysine. And one very important thing is this proline and lysine undergoes hydroxylation. Hydroxylation means attachment of hydroxyl group forming hydroxyproline and hydroxylysine. And vitamin C is required for this reaction. And that is why we say if vitamin C is deficient, it leads to defective collagen formation leading to scurvy. Then type 1 collagen is present in most connective tissues and bone. And fibronectin is a glycoprotein involved in cell adhesion and migration. The questions which have been asked are mainly on proline and lysine. Then moving on to the next slide. Yes, this slide you people should learn by heart. Always remember, because if we talk about biochemistry, first of all, you should know what kind of questions are frequently asked in the exam. Nobody will be telling you to make a cycle. Always the focus is on the deficient enzyme and the disorders associated when a particular enzyme is deficient. And then again, what are the biochemical manifestations of, or what is the clinical orientation if a particular enzyme is absent? So basically we have to focus on the enzymes. So this is a very good slide. You can, take a screenshot of this slide. This you have to learn. Basically, all of you have studied, phenylketonuria is due to the deficiency of, this is a very common enzyme, phenylalanine hydroxylase. Now, this is the enzyme which is responsible for the production of, for the conversion of phenylalanine to tyrosine. Phenylalanine hydroxylase NADPH is required. And BH4 is required, which is not folic acid. Then phenylketonuria type 2, dihydropterin reductase is absent. BH4 is converted into, this is the reaction, into BH2. Always remember this BH4 or BH2 has nothing to do with folic acid. This is not folic acid. Right? So it is dihydropterin reductase. Then albinism is caused due to the deficiency of enzyme tyrosinase, which consists of copper as a cofactor. Then L-captonuria. L-captonuria is the deficient enzyme is homogentisate oxidase. This is a very important question. And alkaptonuria is followed by ochronosis. Benedict's test is positive in this case because homogentisate is a strong reducing agent. Because as you know, the pattern of the question paper is changed a little bit, because last year also it was mentioned most of the clinical based questions will be asked. I will be conducting a session on clinical based questions in a day or so. But this time I'm just preparing you people to, just go through the basics of the subject. Maybe after 2 days, I will be discussing the clinical based questions on this. But always remember, whenever we talk about clinical based questions, try to understand there is always an important point which is hidden in any of the case histories which is there. So this is one of the very important diagnostic feature, ochronosis. There is hyperpigmentation, there is calcification. Okay, ochronosis is there in the pinna and all. So this is what, because it is homogentisate is absent, enzyme to oxidizes, and this homogentisate acid is actually converted into benzoquinone acetates. Then this is a favorite question of the examiners, Hartnupp's disease. Hartnupp's disease is due to the defective tubular reabsorption of tryptophan. Then cystinuria, again very important question. Cystinuria is caused due to, again, defective tubular reabsorption of dibasic acids. And always remember that cystinuria is accompanied by the excretion of 4 amino acids in urine: cysteine, ornithine, lysine, and arginine. So cola is actually due to the defective tubular reabsorption of the dibasic amino acids. Then comes Hawkinsonuria. This is due to the deficiency of enzyme 4-hydroxyphenylpyruvate dioxygenase. So again, I am telling you, this is a very, very important slide. You can just take a screenshot of this. And if you are, sitting with a copy and pen, just note it down. One question will be asked in this. Right? Then moving on to the next slide. Catabolism step of BCAA. Now, all of you know that BCAA stands for branched chain amino acids. So there are so many mnemonics in it. Stands for live or LIV, right? Leucine, isoleucine, and valine. Now these 3 amino acids have a common catabolism. And again, if you have to study, remember the common catabolism step, it is truck hit the ox to death. It undergoes 3 steps, which are common to all the 3 amino acids. But after undergoing these 3 steps, then they follow their carbon skeleton. And on the basis of that carbon skeleton, just now I told you that leucine is ketogenic, isoleucine is keto and gluco, and valine is glucogenic. Now, what are the 3 common reactions? T stands for transamination, amination. O stands for oxidative decarboxylation, and D stands for dehydrogenation. So these are the three common steps. And one very important thing about this is transamination में PLP or vitamin B6 is the coenzyme. Right? In oxidative decarboxylation, TPP is used. And this is FAD-linked dehydrogenation. Means there is a removal of hydrogen by FAD. This was the question which was asked, I guess, 3 or 4 years back, that in oxidative decarboxylation, the coenzyme used is. The coenzyme used is TPP or it is vitamin B1. And deficiency of this decarboxylase or dehydrogenase leads to maple syrup urine disease. It is MSUD. So this is how the branch chain amino acids undergo catabolism. And the mnemonic for this is truck hit the ox to death. 3 common enzymes are there, 3 common steps are there. Transamination, PLP is always required for this. The most frequently asked question. Oxidative decarboxylation, TPP is required. And for dehydrogenation, FAD is required. Right? So this is again a very important slide. Moving on to the next slide. Then now we will be discussing simple important points about different amino acids. See, if you pay attention to all these slides which I'm mentioning to you, the entire syllabus of amino acids or proteins will be finished. So you don't have to go into that much detail before going for the exam. Just go through these last minute revision points. And that is the advantage of discussing these last minute revision points just before 1 week of the exam. Now, whenever we talk about glycine, all of you know that it is a smallest and simplest amino acid. It is optically inactive, very important. Since it is optically inactive, it does not have a chiral carbon atom, or it lacks asymmetric carbon atom. Now, even though glycine is the simplest amino acid, it is required for so many important compounds. Glycine, if it combines with arginine and methionine, it synthesizes creatine, first of all. And this creatine, when this loses a molecule of water, it forms creatinine. Creatinine clearance test is considered to be a very important test for renal function tests. Then glycine, when it combines with succinyl coenzyme A, it is required for the synthesis of heme. In fact, and the enzyme is ALA synthase, which is the most important enzyme in heme synthesis. It forms ALA, amino levulinic acid, which undergoes condensation and the different processes goes on. And this is how it is produced. Right? And then another important point about this is glycine gives C4, C5, N7 of purine nucleus. Because you know the synthesis of purines is de novo, like this. C4, C5, 7, and 7, this portion, synthesized from glycine. Similarly, glycine is a component of Along with the creatine, it is a component of glutathione also, which we discussed just now. So you can very well imagine that glycine being a simplest amino acid, and it is non-essential also. Non-essential means it can be synthesized in our body by serine. Then next, histidine. Now histidine, what all you have to remember? Just now I told you that it is the most stable amino acid at physiological pH. It can serve as the best buffer at pH 7. It can protonate and deprotonate, means it can add addition proton and it can deprotonate at neutral pH. And on decarboxylation, it forms histamine. Decarboxylation means carbon dioxide is removed. And PLP is the coenzyme for decarboxylation. Then another important point about this is excretion of FMGE. It stands for formiminoglutamate acid. Is an indicator of folic acid deficiency. So this FMGE is very important, which is actually formed as a result of degradation of this histidine. Then, and this is a basic amino acid. And it is a semi-essential amino acid. Semi-essential amino acid. Right? Then next comes glutamine. Very, very important. First of all, glutamine is synthesized from glutamic acid. Because this is a carrier of toxic ammonia. And what happens is this glutamic acid binds with this free ammonia, forming glutamine. And this glutamine is transporting and takes it over to the liver so that it can give out this toxic ammonia and it is converted back to glutamate or glutamic acid. Here the enzyme is glutamine synthetase. And here the enzyme is glutaminase. Right? So glutamine is a very important amino acid. In fact, it is a derived amino acid from glutamic acid. And glutamic acid is an acidic amino acid. Right? Then moving on to the next slide, phenylalanine and tyrosine, the most important topic, or in fact, for the most important one for examination point of view. Now see, what happens is phenylalanine and tyrosine. Tyrosine is synthesized from phenylalanine. And tyrosine is a precursor of so many important compounds. Let me just see if this is okay. What happens is this phenylalanine is converted into tyrosine Here the enzyme is phenylalanine hydroxylase and simple NADPH plus H positive is there, which is actually linked to pH2, right? This tyrosine is required for the synthesis of T3 and T4, triiodothyronine and tetraiodothyronine, right? It is required for the synthesis of melanin, which is the skin pigment, and it is required for the synthesis of epinephrine and norepinephrine or the catecholamines. So these are the three very, very important precursors. The question in the exam is asked like this: the precursor of melanin is tyrosine, right? So, and phenylalanine is an essential amino acid. And it is glucogenic as well as ketogenic, right? Then moving on to the next slide. Cysteine. Cysteine and methionine are the two sulfur containing amino acids. Methionine is essential. Cysteine is non-essential. That is why methionine is essential, because it is required for the synthesis of cysteine. That is why cysteine is non-essential. And the most important point about methionine, which you should remember, is what is its active form. Active form is SAM, which is a major methyl donor in the body. For example, synthesis of creatine. And never get confused between cysteine, cysteine, creatine, creatinine. All four are the different entities. Cysteine, two molecules of cysteine combine to form cysteine. And creatine, when gives out a molecule of water, forms creatinine. So you can see what is the difference. Then glutathione. Glutathione ke liye just now I told you, in fact 3-4 slides back, that it is the most important antioxidant. It is required to maintain the integrity of RBCs. Since it is an antioxidant, it is required for the reducing property, right? And then this glutathione is responsible for the transport of certain amino acids across the membranes. And the cycle is known as Miester cycle. Means glutathione by Miester cycle. I won't be discussing the cycle. I'll just discuss superficially. This Miester cycle is responsible for the translocation of amino acids. So it helps in the translocation of all amino acids except proline. This is again very important. Then glutathione is required for detoxification. Means it is responsible for xenobiotic mechanism and, for example, conjugation. In conjugation, the most cited example is degradation of bilirubin, or you can say degradation of heme. The enzyme is glucourinoyl transferase. So it is a very, very important amino acid. Always remember. And that is what is written here in conjugation reactions. And glutathione peroxidase requires selenium as its cofactor. Then tryptophan, a very important, again, a very, loved question of the examiners. Always remember, whenever we talk about proteins or amino acids, the most frequently asked questions are the precursors, because you should know what are the important products synthesized by particular amino acids. Just tryptophan, if we talk about this, it is an essential amino acid, first of all. It is non-polar in nature, and it is required for the synthesis of niacin, which is B3, and serotonin and melatonin. Very, very important. So, because why I have mentioned this thing, because the students get confused between melanin and melatonin. There is a world of difference. If we talk about melanin, melanin precursor is tyrosine, whereas the precursor of melatonin is tryptophan. 60 milligram of tryptophan forms 1 milligram of niacin. So you can very well imagine that if we lack, tryptophan in our diet, we will be more prone to pellagra. And that is what happens. Because people whose staple diet is maize lack tryptophan. So they are more prone to pellagra. Pellagra is again a very, very important question which can be asked in the exam, which is due to the deficiency of niacin, and it is characterized by 3 Ds. What are those 3 Ds? Diarrhea, dementia, and dermatitis. And it gives a Kezar necklace type of feeling. Dryness occurs in the form of a, necklace. That is why it is Kezar necklace. So Kezar necklace is characteristic feature of pellagra, which is due to the deficiency of niacin. And if you look at the precursor, tryptophan is the precursor of niacin. And tryptophan is also known as alpha-amino beta-3 indole propionic acid. Indole group is the most important one in tryptophan. Then comes arginine. Arginine is a basic amino acid, and it is semi-essential. Right? The most important function is it is a precursor of nitric oxide. This reaction I will take in little detail. What happens is arginine is converted into citrulline. And what happens here is nitric oxide is produced. The enzyme is NOS, nitric oxide synthase, and it has a very short half-life. And what is its function? Its function is that it is a vasodilator. It works in accordance with cyclic GMP. Very important. Okay, cyclic GMP is one of the very important second messengers. And it has a direct impact on the smooth muscles. So nitric oxide, the precursor is arginine, and arginine, this is a byproduct, I must say, which is converted into citrulline, forming nitric oxide. And that is what is given in the slide. Then next is alanine. Alanine is a very important glucogenic amino acid. Very, very important. This is the most potent glucogenic amino acid. And it is transported. How it is transported? This alanine is converted into pyruvate by transamination, and the enzyme is ALT, or commonly it is known as serum glutamate pyruvate transaminase. So here what happens is alpha-ketoglutarate is converted into glutamic acid. And since we are talking about transamination, PLP will automatically, by default, it will come. And it forms a Skipp's base. Right? And alanine rises in liver diseases. Always remember. So this is what is alanine, which is considered to be a most important glucogenic amino acid. During starvation, alanine undergoes this, giving energy, because you know it has a tendency to undergo gluconeogenesis, right? Then high yield facts on amino acids. Why I have made this video, I have tried to incorporate each and every damn point which can be asked about amino acids. So if you pay attention to this video, if you pay attention to all the slides which I've been discussing with you, sure and short, some questions will be asked. Right? Because each and every aspect is covered in this. And I have divided my video like that also. Some last minute revision points, some high yield facts on amino acids, right? So all amino acids found in the proteins exist in L form. D and L form, if we talk about, they are what? They are the stereoisomers. Amino acids have two isomers, D and L. And they have only one chiral carbon atom. And it starts from alanine. Glycine does not have any chiral carbon atom, so it is optically inactive and no isomers are produced. Simple. Isoleucine and threonine have two chiral carbon atoms. So the formula for calculating the isomers is 2 raised to power 2, which is 4. Then maximum absorption of light is shown by tryptophan. This is the only aromatic amino acid which absorbs, right? And or this is also a heterocyclic amino acid, just now I have discussed. Then largest amino acid is tryptophan, smallest is glycine. Most acidic amino acid is aspartate, and most basic amino acid is arginine. Most polar amino acid is glutamine, and most non-polar amino acid is phenylalanine, which is also an essential amino acid. Then amino acid with imino group is proline. Amino acid with indole group is tryptophan. Amino acid with guanidium group is arginine. Again, very important, because when we study the qualitative tests on amino acids, it's the amino acid structure which is giving rise to different reactions. 21st amino acid is selenocysteine. Just now I told you standard amino acids are only 20. 21st and 22nd are coded by nonsense codons. But still, Selenocysteine is required as a coenzyme for so many important reactions. Thioredoxin reductase is there, ribonucleotide reductase is there, where it is required, and above all, it has selenium also. So obviously it has a role to play as an antioxidant too. Then 22nd amino acid is pyrrolysine, which is coded by UAG. This is again a stop codon. So that is why we say that 20 standard amino acids are there. And the question, if it comes in the exam, it would be like this only: the 21st amino acid is. The options will be given to you and you have to select. Similarly, the 22nd amino acid is this. Then we come over to, again, a very important topic, which is actually phenylalanine. Now, the reason why I have introduced this slide is because this is the only, you can say, amino acid which has, a rhythm of enzymes. Deficient enzymes leading to different deficiency disorders. And there are certain mnemonics for that. Ab dekho, phenylalanine is converted into tyrosine, right? Tyrosine being an amino acid, it will undergo transamination first, forming para-hydroxyphenylpyruvate, which undergoes dioxygenase activity to form hydroxylase, or I'll write dioxygenase, right? To form homogentisate. Homogentisate will form methyl acetoacetate in the presence of this. And then this methyl acetoacetate will form fumaryl acetoacetate, giving rise to fumaric acid and acetoacetate, showing that it is both glucogenic as well as ketogenic. Now let's look at the disorders. If one is absent, so it is simple phenylketonuria. All of you are familiar with this. Phenylketonuria is due to the deficiency of phenylalanine hydroxylase. Then if transaminase is absent, it leads to type 2, or this is also known as Richner-Hanhart syndrome. Right? If dioxygenase is absent, tyrosinemia type 3 is there, or it is also known as neonatal tyrosinemia. If homogentisate oxidase is absent, L-captonuria is there. And if fumaryl acetoacetate hydrolase is there, it is tyrosinemia type 1. The questions which have been mainly asked in the exam are on phenylketonuria and on homogentisate oxidase. These are the most frequently asked questions in the exam, right? Then this we can discuss later. I'll just show you one very important slide that we can take up in another video, urea cycle. Because urea cycle is again the most favorite topic for the examiners to put in questions. It takes place in 5 steps. Occurs where? Occurs in the liver. And always remember, it occurs in 2 organelles, mitochondria and cytoplasm. Up here, I would just like to bring in one thing in your notice, that always remember this mnemonic hug. Hug you will never forget, isn't it? HUG. Hug is a mnemonic for the processes which takes place in 2 organelles. H is for heme synthesis. U is for urea cycle. And G is for gluconeogenesis. So hug is basically occurring in 2 organelles, mitochondria and cytosol. Okay, this is again a very important question which can be asked in the exam. And in gluconeogenesis, pyruvate carboxylase enzyme is present in the mitochondria. PC. The rest of the enzymes are cytosolic. Right, that is what is written in this. Then rate limiting enzyme is CPS1, which is present in mitochondria because two steps, first and second, takes place in the mitochondria, and third, fourth, and fifth take place in the cytoplasm. Then CPS1, which is the rate limiting enzyme, it is activated by NAG. I'll just write the equation after this. And CPS2 is a cytosolic enzyme for pyrimidine synthesis. Source of ammonia in urine is glutaminase. And one nitrogen atom of urea is derived from NH2 of aspartate. Right, I'll just show you this slide here. Urea is having this structure, NH2, C double bond O, NH2. This carbon comes from carbon dioxide or bicarbonate ions. This comes from aspartate. And this comes from obviously the amino acid. And if we look at the first structure, first step, it is bicarbonate ions combine with ammonia to form carbamoyl phosphate. Right? Here the enzyme is CPS1 and carbamoyl phosphate synthetase 1. So obviously ATP will be required. And another important feature is NAG. N-acetyl glutamate is a positive modulator of this because glutamic acid is actually carrying this toxic ammonia forming glutamine. So this is CPS1, which is present in the mitochondria, and CPS2 is present in the cytosol as it is required for the synthesis of pyrimidines. That is what is written here. So I hope whatever we discussed today is clear to all of you. I have tried to cover each and every topic so nothing is left as such. Right? So this is all for now. In my next video, I will be discussing the last minute revision points on molecular biology. Because molecular biology is the one in which everybody is a little bit scared also. So in my next class, I will be discussing on molecular biology. Then we will touch lipids. So that when you are, ready for your exam, you are all geared up with biochemistry. So don't forget to hook into my next video, which I will be uploading in 2 days or so. So wishing you all the best, study well, because just 1 week is left for your, hardly 1 week is left for your exam. And I would suggest you to again make notes so that you can just go superficially. And always remember, whenever there is a will, there is a way. If you think you can crack the exam, definitely you are going to crack the exam. The only thing is that your strategy should be such that everything should be crystal clear in your mind. This is very, very important. You should be confident on what you are writing. And always remember, in biochemistry, all the standard type of questions have been asked. And I'll try to keep a session on, case histories also. Because if a question on case history comes, then definitely you should be able to answer it. So wishing you all the best. Let's wind up this. Have a lovely weekend. Bye-bye.